Hello, hi, welcome to NEET Savers. This is your KK sir. Yes, sir. In this session, we are going to discuss the previous questions which were asked from the topic fertilization and implantation. After this, we are going to see some of the previous questions again in the next session. Right, let us switch into our session. Fertilization and implantation. Already we discussed in our class so many things we have discussed about fertilization and implantation. Now let us see what were the questions asked, right? Here is the first question given below two statements. One is the assertion, second one is the reason. Let us say 74th question. Here it is. Right, assertion is endometrium is necessary for implantation of blastocyst. Yes, we know that the innermost layer of the uterus is known as endometrium, which is very essential for the embryonic growth. That is, implantation is nothing but the blastocyst is going to be transported from the fallopian tube into the uterus. It is going to be attached with the help of the trophoblast, right? So, this attachment we know that is the implantation. The endometrium is very essential for the implantation. So that the assertion which is given here is right one. That means the first assertion, the assertion is right. And let us see the second one, that is the reason. In the absence of fertilization, if the fertilization doesn't take place, what happens? The endometrium is going to shed off from the uterus walls, right? And it will be sent out through the birth canal. So that is known as, we know that that is the menstrual flow. In the absence of fertilization, the corpus luteum degenerates. Yes, absolutely. Here, if the fertilization takes place, what happens? The corpus luteum will be there in the ovary itself. That corpus luteum acts as the temporary secretory gland or temporary endocrine gland. This particular corpus luteum, we know that it's going to generate, it's going to produce the progesterone, right? The progesterone, which is very essential for growth of endometrium. And this particular corpus luteum, it also secretes one more hormone, that is relaxin, which is not given in our textbook. Relaxin also secreted by corpus luteum. So this is very useful when the time of the delivery. So again, during this particular gestation period, the pelvic ligaments to be loosened. So for that, this relaxin should be there, right? This particular relaxin is going to be produced by the corpus luteum. So corpus luteum is a structure. It's a structure of the Graafian follicle after release of ovum from the Graafian follicle. So the remaining structure we call it as a corpus luteum, that we know. So in the absence of fertilization, the corpus luteum degenerates. Yes, it is right. That causes the disintegration of endometrium. So as a result, what happened? The menstrual flow continues. The endometrium itself, the endometrium along with the blood vessels of maternal blood vessels, these all things, they come out as a menstruation, right? So this is also right. Endometrium is necessary for implantation of blastocyst. Yes, it is the right one. And in the absence of fertilization, so it is a continuation. This is the continuous statement for this one, right? If the fertilization occurs, this happens, right? If the fertilization doesn't take place, this corpus luteum degenerates. That causes disintegration of the endometrium. So this is the right one, first one, and this is the correct explanation for the A1. Right here, let us see then. A is true, but R is false. So this is the wrong option. A is false, but R is true. But A also here, right one. So A also true here. Both the A and R are true, and R is the correct explanation of A. Now our option is C. C is the right option. So you remember that this corpus luteum secretes not only the progesterone, it also secretes relaxin hormone, which acts on the ligaments of the pelvic bones, and these ligaments are going to be loosened when the mother is bearing the baby and the womb becomes larger and larger when the gestational period increasing gradually. That time ligaments has to be loosened, and at the time of delivery also these ligament has to be stretched more, right? Further, this relaxin hormone is going to act on that. Now second question, 75th question we are going to see. Receptors for sperm binding in mammalian are present on, right? Here we have to observe one thing. The sperm is going to attach to which part of the ovum? Sperm is going to attach the zona pellucida, that is the outermost layer of the ovum, right? Zona pellucida, which is covering outermost of the ovum. So first thing is that this particular sperm is going to attach the zona pellucida. From there, the acrosome, what is that? The cap-like structure which is present on the head of the sperm, it is going to release some kinds of hormones, right? Like acrosine, hyaluronidase. These are digestive enzymes which are going to digest the zona pellucida in order to enter the sperm into the ovum. Right, first thing is that the sperm is going to attach the zona pellucida of the ovum. So, let us see that receptors for the sperm binding. So, if at all, if a sperm wants to attach the particular part of the ovum, it should have some kind of receptors on it, receptors, right. So, these receptors are present in the zona pellucida where the sperms are going to be attached. Only one sperm is going to, not only the one sperm actually, so many sperms are going to be attached, but the first sperm which attaches to the zona pellucida, that only enters into the, sperm's head only enters into the cytoplasm of that particular ovum. After that, cortical reaction takes place. Ultimately, the outermost layer which is present around the ovum, it is going to become very tough. So, in order to block the other entry of the sperms, other sperms entry into the ovum. So, that is a polyspermy. If polyspermy occurs, what happens? The number of chromosomes going to be increased in the zygote, which is not resulting a human being, right. So, here the zona pellucida is the right answer. And next, extrusion of second polar body from egg occurs. So, extrusion means here the release or formation of the second polar body from egg occurs. When does it occurs? When the sperm touches the ovum. When the sperm touches the ovum, when the sperm attaches the zona pellucida of the ovum, that time only second meiosis division takes place. As a result, from the secondary oocyte, secondary polar body as well as ootid are going to be formed. That particular ootid further it is going to be formed as the matured ovum, right. Right, here we will see simultaneously with the first cleavage, not that one. So, first cleavage occurs in the embryonic stage itself, right, and after entry of the sperm, but before the fertilization. Before the fertilization itself, after entry of the sperm, but before the fertilization, after the fertilization. See, after the fertilization, it does not take place. Why? Because, so meiosis 2 has to be taken place before the fertilization itself. Then only we will get the ovum, right? Then only we will get the oocyte. So, this particular phenomenon, it must take before the fertilization itself. Now, this is the wrong answer and this is the wrong answer, and before the entry of sperm into the ovum. No. So, the ovum is not going to be initiated till the sperm touches the ovum. Now, this is also false answer. Now, the right option is after entry of the sperm. So, this is the, here, this is the ovum when it is touching, when the sperm is touching, So, here the second meiosis division takes place. Keep it in the mind. The second meiotic division takes place when the sperm touches the zona pellucida. Then the ovum is going to be the secondary oocyte which is inside here, that is going to be initiated, and the meiosis 2 takes place. As a result, second polar body as well as the ootid is going to be formed. So, now the answer is this one, right. And switch on to the next one, that is capacitation occurs in. What is a capacitation? We don't come across in our textbook, right. We don't come across this particular word, this particular term in our textbook because it was asked in the 2017. So, it is somewhat which is not given in our textbooks. Capacitation is nothing but activation of the sperm. See, the sperm moves slowly, very slowly, like 0.2 mm per minute, it moves very slowly. So, if it moves like that, it can't reach the ovum. So, it has to be induced, it has to be motivated, it has to be it has to be activated. So, actived sperm moves very fastly. So, this activation occurs, or else this initiation of this particular activation is done by the secretions which are secreted from the female reproductive tract. So, then this sperm will become very active. So, the sperm becoming active is nothing but capacitation. See, this capacitation occurs in epidermis, not this one, vasa deferens, not this one, female reproductive tract. So, in the female reproductive tract only, it is going to secrete some kind of secretions. By these secretions only the capacitation takes place. Right? Read it as these three belongs to male reproductive system, but capacitation generally occurs in the female reproductive tract by the secretions of the female reproductive tract. Keep it in the mind, right? This is the capacitation. Now, we will see the next one. fertilization in humans is practically feasible only if. See, here you have to observe each and every word, The ovum and sperms are transported simultaneously to ampullary isthmic junction of the cervix. So, you should not be confused and you should not be overexcited. If one excites, what happens here? Immediately, without reading all these options, you are going to tick this is the right answer. But let us see whether it is right or wrong. The sperms are transported into cervix within the 48 hours of release of the ovum in the uterus. The sperms are transported into the vagina just after the release of the ovum in the fallopian tube. The ovum and sperms are transported simultaneously to ampullary isthmic junction of the fallopian tube. See here, if you read carefully, this A and C, A and D options looking very similar, If you differentiate, then only you can find out the right answer. When do you differentiate? You should go through it very carefully and very clearly. Right here, see here, simultaneous to ampullary isthmic junction of the cervix, but there is no such region in the cervix region, right? So, is there any ampullary isthmic junction in the cervix? No such part is present in the cervix. It's a narrower part of the uterus which has the cervical canal in it. That's it. Only cervical canal is present in cervix. So, this particular, what is that? Ampulla, isthmus as well as infundibulum. These three are the parts of fallopian tube, right? So, here this is the right answer. The ovum and sperms are transported simultaneously to the ampullary isthmic junction of the fallopian tube, right? You have to find out the right answer. So, this is the right one. So, it was asked in the NEET 2016. And let us see the 79th one. In human females, meiosis II is not completed until. Now, see here how many questions were asked in this particular topic only. Here, meiosis II. So, this particular oogenesis process we should know very clearly, oogenesis as well as the fertilization process. Depending on that, here the questions were asked. Same, this kind of question was asked before, right? So, uterine implantation is a birth, puberty at the time of fertilization. You must read it very carefully. In human females, meiosis II, he is asking meiosis II, not meiosis 1. So, if it was meiosis 1, what is that? So, before the birth itself, these primary oocytes are going to be stopped at the stage of prophase 1, right? But here he is asking, the examiner asking meiosis II is not completed until, not the puberty here. So, until the fertilization is the right answer. So, already we discussed just before. So, when the sperm is attaching to the particular receptors which are present on the zona pellucida, when this particular sperm is going to attach those receptors, then this meiosis II is going to be initiated in the secondary oocyte, right? Then only the secondary meiosis, that means that what is that? That is meiosis II is going to be taking place. So, when the sperm touches, that is the fertilization process. At the time of the fertilization process, this particular thing is going to be taken place, right? So, what happens after the meiosis II? The second polar body as well as oocyte is going to be formed, right? So, this is the question here, the fertilization. And here the capacitation refers to change in, right? So, already we discussed that ovum after the fertilization, not at all the capacitation, this particular term or word which is not at all related to ovum, it is completely related to sperm. Ovum after fertilization, not this one, and ovum before the fertilization, this is not answer, this is the not right answer, right? Sperm after fertilization, see here, before fertilization itself the sperm has to be activated, right? So, after the fertilization, what happened? The sperm is going to shed off from the oval surface. Right here, the sperm before the fertilization. Now, this is the right answer. The sperm has to be activated before the fertilization itself. Then only it enters into the ovum. It moves very fastly, it moves very vigorously in order to reach the ovum. So, that particular activation of the sperm is known as capacitation, right? And let us discuss next one. In our society, women are blamed for the producing female children. Choose the correct answer for the sex determination in the humans. Right? This particular question asked from the sex determination in the human beings. Due to some defect like aspermia in man. Aspermia means that is nothing but there is no enough sperms present in the male in order to fertilize the ovum. That is known as aspermia. Aspermia is A means without, sperms means spermia. Due to the genetic makeup of the particular sperm which fertilize the egg, due to the genetic makeup of the egg, due to some defect in the woman. Here if you observe that, so there are two kinds of sperms are produced in the man, right? So one sperm, generally one kind of sperm, they contain 22 plus X chromosome, and 50% of the sperms, they contain 22 plus X chromosome. How many sperms are produced in an ejaculation? 200 to 300 millions of sperms are being produced in an ejaculation. In them, 50% of them, they have the genetic makeup with the 22 autosomes with the one allosome, that is the X, and 22 autosomes with the Y chromosome. 50% of the sperms are like this, right? 50% of the sperms, their genetic makeup is nothing but, so what kind of chromosomes are present there? So what kind of chromosomes are present in a sperm? So that is the genetic makeup of a sperm. If you observe the genetic makeup of ovum, all the ova they contain 22 plus X chromosome only. Only one kind of ova are released in the human being. Whereas when we coming to sperm genetic makeup, so there are 50% of the sperms alike and the 50% of the sperms alike, right? See here, 50% of the sperm, they contain 22 plus X chromosomes, right? And 50% of the sperms, they contain 22 plus Y chromosome. When this particular, see here, entire the sex determination is depending on the father itself, because if the sperm which is containing the Y chromosome, if it fuses with the ovum, then it leads to the formation of the male baby, right? So if the sperm it contains X chromosome, if this sperm fuses or fertilizes the ovum, what happens? The resultant baby is female, right? So in this way, this particular sex determination process is depending on the sperm, the type of sperm which is going to fertilize the ovum. Here the right answer is due to the genetic makeup of the particular sperm which fertilize the egg. If the sperm contains Y chromosome, then it leads to the formation of the male baby, if at all. Right here, first you observe the diagram first. So this was given very clearly. See here, you can see the inner cell mass, it contains stem cells, and outermost layer is trophoblast. From the trophoblast, extra embryonic members are going to be developed, and from here from the stem cells, or this is the cell mass, inner cell mass. So this particular inner cell mass is going to be developed as embryo. That is a clear one. And here you can see the space, this is the empty space. If a particular stage of the development of the embryo, if it has the empty space inside, then we call it as a cyst. So cyst means here the empty space, it's a ball like structure which has the empty space inside it, then we call it as a cyst, right? So this is the blastocyst, we can clearly see that this is the blastocyst. In between this particular cell mass as well as the trophoblast, we can find some more inner cells which belongs to the trophoblast. So these are the inner cells of the trophoblast. Generally we call it as a robust layer. So from the robust layers only, these extra embryonic membranes are going to be developed. So what are the extra embryonic membranes? Generally this is allantois, chorion, as well as this one. So amnion. So these three are extra embryonic membrane. Extra means outer. So these membranes are going to wrap around the embryo. So they are present around the embryo. So that we are calling them as the extra embryonic membranes. So these are the robust membrane, right? And inner cell mass, which are further going to be divided into two. So when it is in the two layer stage, we generally call it as embryonic disc. So one is epiblast and second one is the hypoblast. So from the hypoblast, what is that? Primary yolk sac is going to be developed, and from the epiblast there are so many derms are going to be formed. One is ectoderm, mesoderm, and third one is endoderm. So from these three germinal layers, all the organs of the embryo is going to be developed from these three, right. So, this is all about this particular stage, right. We'll see this, we'll see the question. If at all first, if there is any diagram is given in the question, first you observe the diagram and first you try to understand that, and first you must recognize what kind of diagram it is, right. So, here this is very, we know that this very clearly, we know that that is the blastocyst, right. Identify the human developmental stage shown as well as, so first we have to identify this one shown as well as the related right place of its occurrence. Where do you find the blastocyst? So, it moves from the fallopian tube and is going to implant in the uterus, right. So, a normal pregnant woman, see here, a normal pregnant woman, you have to observe very carefully each and every, see how he is playing the logic. So, normal pregnant woman, he said. So, there is a chance of implantation of this particular blastocyst in sometimes it is going to be attached in the fallopian tube itself. It is going to become embryo in the fallopian tube itself. So, that here the word which is used here, a normal pregnant woman, the normal pregnant woman in the sense the blastocyst which is given here, it must implant in the uterus itself. That is what this particular word is used here, right. Right, let us see that. In the normal pregnant woman and select the right option for two together. See here, this is late Marla, no, this is not at all late Marla, and this is not at all blastula because there is a cyst, there is an empty space. So, generally blastula means completely it is a solid one, right. So, blastocyst and uterine wall. So, this is the right option here. 8-celled Marla, here not at all 8 cells are present here. 8-celled Marla is also wrong option. Now, this is the blastocyst, it is going to attach the endometrium of the uterus. Now, the option C is the right answer. And let us see this one, next one, the 2011 question. What happens during the fertilization of the human after many sperms reach close to the ovum? So, what happens generally, so many millions of the sperms are released in the female genital tract, right. So, that particular process, insemination, we call it as insemination. So many sperms, they try to reach the ovum to fertilize it. So many sperms are going to be attached to the surface of the ovum, but only one sperm is going to be enter into the ovum when it first touches the zona pellucida, as well as the so many enzymes are released from the acrosome. That particular sperm which is releasing the enzymes, which is digesting the zona pellucida of the ovum, that particular sperm only going to enter into the ovum and it is going to fertilize the ovum, and after entering into the ovum, the entire outermost is going to be hardened to prevent the polyembryony, that means polyfertilization. Right, let us see the options here. Secretions of acrosome helps one sperm enter cytoplasm of ovum through the zona pellucida. This particular option is looking right. All sperms except the one nearest to the ovum lose their tails. He is not saying it enters into the ovum, as well as here he is saying, the examiner is saying, all the sperms except one sperm nearest to the ovum lose their tails. All the sperm, they are going to lose their tails. This is also not right answer. Cells of corona radiator trap all the sperm except one. So here the zona pellucida, it's going to play a vital role, right? Now he is giving corona radiator. No, only two sperms nearest the ovum penetrate zona pellucida. If the two sperms enters into the ovum, what happens? The number of chromosomes are going to be increased than the normal, right? The sperm contains 23. Again, one more sperm, he is saying 2. So the ovum contains 23 chromosomes. What happens here? The number of chromosomes become 69. So this is not at all like an human being. So generally they contain 46 chromosomes. If two sperms enter, the total number of chromosomes become 69 and the complete zygote becomes triploid. So this is also wrong answer. So ultimately we can say that the first option which is given here, secretions of acrosome. What are the secretions from the acrosome? Generally acrosin, hyaluronidase and corona penetrating enzymes. Corona penetrating, these three are released from the acrosin when they bind to the particular receptor present on the zona pellucida of the ovum. So then only the acrosomal reactions takes place, right? So this is the right answer. A is the, and this is the 84th one. The second maturation division. So the second maturation division, that is meiosis 2. See here, this is the third time they are asking from this particular phenomenon. The second maturation division of the mammalian ovum occurs shortly after ovulation before the ovum makes entry into the fallopian tube. No, this is wrong answer. Already we discussed so many times this about this one. Until after the ovum has been penetrating by the sperm. Until after the ovum has been penetrated by a sperm, right? Until the nucleus of the sperm has fused with that of the ovum. Until the nucleus of the sperms has fused with that of the ovum. In the Graafian follicle following the first maturation division. No, this is the wrong answer and this is the wrong answer. So we have to check these both statements together. Once again we'll go through those two statements. Until after the ovum has been penetrated by a sperm. See here, until after the ovum has been penetrated by a sperm. Until the nucleus of the sperm has fused with that of the ovum. No, this is the wrong answer. So here this is the right answer. When the sperm is trying to penetrate into the ovum, the secondary oocyte is going to be initiated and the secondary meiosis division takes place over there. Now this is the right answer. So here until the nucleus of the sperm, he given until the nucleus of the sperm. So this is not the right, this is the right answer. Now let us check next one. Which one of the following statement about morula in humans is correct? He is saying about the morula. So what is the morula? What is the morula? Zygote. When the zygote is formed in the ampullary ischemic junction, what happens? It is rolling down towards uterus, right? When it is moving towards the uterus, what happens? So many cleavages happens in the particular zygote. As a result, the number of cells are going to be increased and generally this mitotic division takes place, right? So this we know that we call it as cleavages, and the cells which are formed by this cleavage we call them as blastomeres. So generally in the 16 cell stage we call it as morula, right? So we are going to see about the what are the right statement, the following statements about the morula. It has almost equal quantity of cytoplasm. See here, I said already when we were talking about this particular phenomenon. So it has almost equal quantity of cytoplasm as an uncleaved zygote, but much more DNA. See here, this is the zygote, right? This is the zygote. Say for example, there is a 1 gram of DNA, 1 gram, assume that, so no such grams are there, 1 gram of DNA and 10 grams of cytoplasm, 10 grams of cytoplasm. So when it is dividing into two cells, what happened? The number of cells are increased, but the cell size is decreased. The cell size is here this much larger, this much big, and if you observe after the first mitotic division, the cells which are formed by this particular first mitotic division, the 2 cells are formed and here the DNA is increased 2 grams, okay. The cytoplasm is going to be decreased, right? So no, not actually, the cytoplasm is not going to be decreased because here 10 grams, here 5 grams of cytoplasm, in this cell 5 grams of cytoplasm. So ultimately cytoplasm quantity is being maintained constantly and the number of cells are being increased and the size of cell is going to be decreased, and here if you observe the genetic material of the daughter cells, this particular genetic material, quantity of the genetic material is going to be increased. See here again we will see one more mitotic division, that is the 4 cells. So 4 cells are going to be formed, right? From this cell 2 cells, from this cell again 2 cells. Again see here you can see the quantity of the DNA is going to be increased by 4 grams, whereas the cytoplasm, see if the size of the zygote is increased, if the size of the cells are increased, then cytoplasm is going to be increased. But here if you observe carefully, in our textbook also you observe the diagrams carefully. See here, these are the daughter cells which are not bigger than the mother cells. They are smaller than the mother cells. Ultimately, the cytoplasm remains as it is. So here the 5 grams, in the sense here in this one 2.5 grams, 2.5 grams, 2.5, 2.5. Ultimately, the total quantity of the cytoplasm is 10 grams only, right? In this way, the number of cells are increased, the size of the cells is going to be decreased, and this particular quantity of the cytoplasm is going to be maintained constantly, and here the genetic material is going to be increased in its quantity. These are the different changes taking place in the marla. Let us check which one is the right one. It has almost equal quantity of cytoplasm as an uncleaved zygote. This is not yet cleaved. So this is not yet undergone mitosis process, right? This is uncleaved zygote, but much more DNA. So assume that this is a marla. So it has the much more DNA. It has a much more DNA when comparing with the zygote, right? It has only 1 gram of DNA, but here the 4 grams of DNA of present, 4 grams of DNA, right? If it is enters into the 16 cell, what happens? 16 grams of DNA, the quantity of the DNA is being increased gradually. So the marla stage has the more amount of the genetic material when comparing with uncleaved zygote, this uncleaved zygote, right? So it has almost equal quantity of the cytoplasm, right? Cytoplasm quantity is not being changed here, right? So equal quantity of cytoplasm as an uncleaved zygote, but much more DNA. Yes, absolutely correct statement this one, right? It has far less cytoplasm. It doesn't have the lesser cytoplasm than the uncleaved zygote. So as well as the less DNA. So this is also wrong option. And we'll check next one. What are the options given here? It has more or less equal quantity of the cytoplasm and DNA as in uncleaved zygote. So DNA quantity is not being changed as well as the cytoplasm quantity is not being changed in this option that is given like this. But here the quantity of DNA is being increased. Remember that one. So the quantity of the DNA is increased in the marula. So this is also wrong statement. What happened here? What the statement given here? It has more or less equal quantity of the cytoplasm and DNA. In the sense, whatever the thing is applying here, the cytoplasm, the same thing is applying to the DNA here also, right? So this is also wrong option. It has more cytoplasm and more DNA than an uncleaved zygote. So here we do not see any kind of change in the quantity of the cytoplasm, but the statement has given marla has more cytoplasm and more DNA than uncleaved zygote. This is also wrong statement because the quantity of the cytoplasm is being maintained constantly, whereas the quantity of the DNA is becoming more and more when comparing with the zygote in case of the marla, right? So this is also wrong statement. So ultimately the A answer is right. The correct answer is the right. A1, the first one is. In the human females, the blastocyst. Now the marla stage is over. Now he is focusing on the blastocyst. Let us see that. Forms placenta even before implantation. So before implantation itself, the blastocyst was going to form placenta, he is saying. So ultimately this is wrong one. So after the implantation around 3rd month or after 12 weeks, around 12 weeks like that. So the outermost layer which is present, which we call it as a trophoblast, finger like projections are going to be developed from the trophoblast, which we call them as chorionic villi. So around these chorionic villi, the uterus tissue is going to be very close. The uterus tissue is going to wrap around this particular chorionic villi, as well as the maternal blood also present around these chorionic villi. All these together, this particular functional structure we call it as a placenta. So placenta is going to release so many hormones. One is HCG and HPL as well as estrogen, progesterone. These hormones are released by the placenta. Right now we'll see the second option, that is gets implanted into uterus 3 days after ovulation. Gets implanted into uterus 3 days after ovulation. Gets nutrition from uterine endometrial secretions only after the implantation. Gets implanted in endometrium by the trophoblast cells, right? This is the correct option. So after the formation of the trophoblast, these trophoblast cells, we know that they are going to release, they are going to form some kind of structures. So this particular trophoblast, when it has a trophoblast around it, then only it's going to implant in the uteral wall, in the endometrium, right? So this is the correct option. So here get nutrition from the uterine endometrial. When does it get the nutrients from the endometrial layer? When the complete placenta is formed, then only this particular placenta, it's going to get the nutrients from the maternal blood and it's going to supply to the embryo through its umbilical cord. So leave it that, leave this one, these three are wrong and this is the correct answer. And let's check the next one, 87th one. A change in the amount of yolk. What is the yolk? Just it's like a yellow colored part in an egg is called as yolk. A change in the amount of the yolk and its distribution in the egg will affect. See here, assume that this is the egg, this is the egg. If we have more amount of yolk, what happens? The cleavages takes place here only where the yolk is not going to be present. Where the yolk is not present, the cleavages takes place partially or a part of the egg only. Because here this particular yolk which is present in the egg, it prevents the cleavages in this particular region. As the yolk is present, so there is no cleavage in that particular region. So if the yolk, where the yolk is not present, that in that particular region only the cleavage generally takes place. So it's preventing, it's blocking the cleavages of a zygote, right, of a particular fertilized egg. So our eggs are, human eggs are alethal eggs. Alethal in the sense no yolk or very little quantity of the yolk is present. Such eggs are known as alethal eggs. If egg is alethal, that means no yolk is present. What happens? Entire zygote is going to be cleaved, right? So if the entire part of the zygote is going to be cleaved, and that particular cleavage is known as holoblastic cleavage. See here, if the portion of the zygote is undergoing cleavage process, that particular cleavage process meroblastic cleavage. If the entire portion of the zygote is undergoing the cleavage process, then this particular process is known as holoblastic, right? In human being generally our zygote undergoes holoblastic cleavage. Say if you take the hen egg, what happens here? The meroblastic cleavage takes place, right? This is the process actually happened. Here we'll see the question actually. A change in the amount of the yolk and its distribution in the egg will affect pattern of cleavage, right? Exactly right, this one. Number of blastomere produced, this is wrong answer. Fertilization, wrong answer. Formation of the zygote. So these three are wrong answers and this is the right answer. So if the yolk is present, then cleavage is particularly restricted to one region where the yolk is not present. So it's going to affect the cleavage process. Now actually some of the questions were not actually given, not even given in our textbook. So because these were asked a few years back. So maybe this particular topic were there, maybe we can't say that. So this is the question which was asked in the 2009, maybe in that year. So there may be this particular topic in the syllabus, right? So gray crescent is the area, at the point of entry of the sperm into the ovum. So this is the reception cone, we call it as, just opposite to the site of entry of the sperm into the ovum. So this is the right answer. So gray crescent is the just opposite to the site of the entry of the sperm. So this was the question asked in 2005. So it may be, it was there, it may be there in the textbook. So we don't know. And let us see the 89th question. What is the true for cleavage? Size of embryo increases, size of cells decreases. See here, size of the cells is decreases, size of cell increases, size of embryo decreases. So forget about this embryo and let us check these two statements. Size of cells decreases, already we discussed. When the zygote undergo cleavage process, the total number of cells are increased, but the size of cells are going to be decreased, right? So now the size of cells is going to be decreased. So that is the correct statement regarding the cleavage process. already just now, just before we discussed by drawing pictures here. And 90th one, blastopore is the pore of, blastopore is the pore of. Here this is one, the blastopore is the pore of archenteron, blastocele, coelom and alimentary canal. So alimentary canal is the digestive system. After the birth in our adults also, or else after the birth in the children also, we call it as alimentary canal. This is the digestive tract. So at the development of alimentary canal in the embryonic stage, we call it as archenteron. So primitive gut, we can call it as primitive gut. So this particular archenteron further it is going to be developed as alimentary canal. So this is the embryonic stage archenteron, and further it is going to be developed as alimentary canal. So blastocele is nothing but the empty space which is present inside the blastula is blastocyst. We call it as blastocele. So in the development stage, what happens? This blastocele is completely occupied by the cells. So by the cells which are incoming inside the blastocele. So complete blastocele going to be disappear after the completion of the gastrulation. So that is the process. Leave it. Blastopore remains same. So this blastopore is the pore of blastocele, right? So blastopore is the pore of archenteron or coelom. So coelom also you just forget about this one. So now here blastopore is the pore of archenteron. This is the primitive gut of the embryo, right? And let us see the 91 question. 91st question. Fertilizin. See here, acrocin is released by the acrosome of the sperm. Fertilizin is a substance which is released by the egg to attract the sperm. The ovum is going to release the fertilizin. This particular word, this particular term also not given in our NCERT textbook. Fertilizin is a chemical substance produced from polar bodies, middle piece of the sperm, mature eggs and acrosome. So acrosome, leave it about that because this is entirely related to sperm. So this particular which is released by the mature egg in order to attract the sperms, right? So fertilizin is a chemical, or it's a substance which is released by matured eggs, right? Remember this one, this particular one. So actually it was asked in 1997, 1991. So it may be there present in those books. Again, we'll see the 92nd one. So in human beings, the eggs are just now we discussed, no yolk is present, or very less amount, very minute amount of yolk is present in human eggs. So that ultimately we call it as A-less cell. A means without. A means without. So without yolk, the eggs are called A-less cell eggs, right? Option is A-less cell eggs is the right answer. Right, 93rd one we'll see. In an egg, the type of cleavage is determined by. So what kind of cleavage it is happening in the zygote? So is it meroblastic? Is it holoblastic? Depending on which factor, the type of cleavage is going to be determined. Already I said the distribution of yolk present in the egg, it's going to determine the type of cleavage. If more amount of yolk is present, what happens? It undergo meroblastic division. That is, the zygote is going to be divided partially. It undergoes partially, right? If no yolk is present, what happens? The entire zygote is going to be undergo cleavage process. That particular process is known as holoblastic cleavage. The amount and distribution of yolk, this is the right answer. The number of egg membrane, not this one. So the shape and size of the sperm, not this one. The size and location of the nucleus, so not this one also. The nucleus not at all is going to affect the cleavage process. Only the amount of the yolk and the position, the amount and distribution of the yolk is going to affect the cleavage process. And what type of cleavage should go in that particular zygote, the amount of the yolk is going to determine that particular one. What is the true about the cleavage in the fertilized egg in human? So observe the question. What is true about the cleavage in the fertilized egg in humans? It starts while the egg is in fallopian tube. while the egg is in fallopian tube. It starts when the egg reaches the uterus. When egg reaches the uterus, it is meroblastic, not at all meroblastic. Our cleavage is holoblastic. The entire zygote undergo the cleavage process. It is identical to the normal mitosis, not at all, not at all. This is not at all general and simple mitosis process, right? Now, it starts while the egg is in fallopian tube only. The cleavage process takes place when the fertilized egg, it is moving towards, it is rolling down through the fallopian tube. Generally, the number of cells are going to be increased, right? In the sense, that means the cleavage process starts when the zygote is formed in the ampullary isthmus junction. So, generally it moves very slowly towards the uterus. When it is moving towards the uterus, that time only the entire cleavage process, not entire, so up to the morula and blastula is going to be formed when it is rolling down through the fallopian tube. Now, it starts while, it means here the cleavage process starts while the egg is in fallopian tube, right? This is the right answer. So, here if you look at the second statement, it starts when the egg reaches the uterus. When egg reaches the uterus, this is the entirely wrong answer, because till it reaches the uterus, it is in the blastocyst stage, right? So, it reaches the blastocyst stage, then only it is going to implant in the uterus. Now, this is the wrong statement, you can simply say that. Now, we will see the 95th one. Termination of gastrulation is indicated by termination of gastrulation. Already I said just before we discussed that. Termination of gastrulation process. What is the gastrulation process? There is a primitive streak formed in the zygote. So many cells they are going to enter inside the gastrula, and they are going to completely occupy the empty space which is present inside the ball-like structure. So, after occupying all this empty space inside the gastrula, the gastrulation stage is going to be stopped. So, that is the actually thing happened there. Termination of gastrulation is indicated by obliteration of blastocele. Blastocele is nothing but the empty space which is present in the blastula is known as blastocele. Obliteration of archenteron. So, archenteron is not going to be obliterated. Obliterated means complete occupying. See, for example, there is an empty space. See, from here something is some cells are coming, and this empty space, this particular empty space is being occupied these cells. So, this particular process is known as obliteration, right? If what happens, obliteration of archenteron, if obliteration of archenterons happen, what happens? Entire gut is going to be closed, right? But here it is not happening. So, obliteration, so occupying of the cells into the, or else entering of so many cells into the blastocele. So, blastocele is going to be closed. So, if blastocele is going to be closed, then the gastrulation process is going to be completed, right? And closure of blastopore. If blastopore closes, what happened? We don't have mouth. We would not have mouth if the blastopore is going to be closed. Closure of neural tube. If neural tube closes, what happens? If neural tube is going to be closed, if the neural tube is closed, what happens? CF is not present in the inside the neural tube. So, this is the wrong. So, ultimately the right answer is obliteration of the blastocele is the right answer. It indicates the termination of the gastrular phase. In telolesthal egg, telo means telo, wherever you come across this word, telo means tail or end, or else a side. Telo means telolesthal egg, the yolk is found all over the egg, on one side, both the side, center. So, this is the on one side. So, see, if the egg is like this, the yolk is present in this region. So, just the yolk is present one side of the egg. So, this particular egg is known as telolesthal egg. So, we do not have the types of eggs, microlesthal, telolesthal, megalesthal, these are the different types of the eggs, but we do not have in our syllabus. And next we will check the acrosome reaction in sperm is triggered by. Acrosome reaction in sperm is triggered by capacitation, influx of sodium, release of lysine, release of fertilizin. So, here the capacitation, actually this particular acrosomal reaction, that means So capacitation means the sperms are going to be activated before it reaches the egg. Now this is not right answer. And release of lysine, when release of lysine, so when does the sperm release the lysine? When it attaches to the zona pellucida. And influx of sodium release of fertilizing. So not this one. Here we have to check the answers C and B. Release of lysine. So when does the lysine releases? So there should be some kind of reactions in the sperm body. So then only they are going to release the lysines. Lysine means, so they are the enzymes which are present in the acrosome. So that is the lysine. And here influx of sodium as well as calcium ions also. Influx of sodium ions as well calcium ions, they move inside the sperm's body. So as a result, the acrosomal reactions takes place. That means the acrosome is going to release some kind of enzyme which are going to react, or which are going to digest the zona pellucida in order to enter into the ova. So here this is the right answer, influx of sodium as well as calcium also, but calcium is not given here. So it was the question of 1993. So many years back question this one is. And meroblastic cleavage is a division which is, already I said, so generally if the yolk is distributed in the egg, so this particular cleavage process restricted to particular one area. So it is very confined to one area where the yolk is not present. So because the cleavage process is particularly to a particular portion of the egg, so the portion of the zygote. So horizontal, total, spiral, partial or parietal. So this is the right answer. The partial cleavage of a zygote is known as meroblastic cleavage. And blastopore is opening of future anterior end of embryo found in blastula, opening of gastrocele and opening of neural tube. Here gastrocele, opening of the gastrocele is nothing but blastopore, right? Right, next question is, during the cleavage what is true about the cells? During the cleavage, what is true about cells? Nucleocytoplasmic ratio remains unchanged. Observe the statement, what he is saying, nucleocytoplasmic ratio, that is nucleoplasm means here the DNA, nucleo means, and cytoplasmic ratio is remains unchanged. This is wrong answer, because cytoplasm quantity remains same, but the nucleo, that means the genetic material, the genetic material quantity is going to be increased. So here it is both the quantities unchanged, the option is wrong. Size does not increase, there is less consumption of oxygen. The division is like meiosis, the division not at all meiosis. So we can cross it, and we can cross this one. Size does not increase, see, if this is the zygote and this is the 16 cell stage, the size of the zygote is not going to be increased. If the size is like this, here also in the morula stage also size is not going to be increased. So this is the right option here. Okay, and next, freshly released human egg, they contain generally 22 autosomes with X chromosome. Only one X chromosome is present, in the egg. So this is the right answer about the freshly released human egg. Sperm and egg nuclei fuse due to, so there is attraction between these two cytoplasms, the cytoplasm of the egg as well as cytoplasm of the sperms. There is a particular attraction between these two cytoplasms. These two cytoplasms are generally, we call them as protoplast. Base pairing of their DNA and RNA, formation of hydrogen bonds, mutual attraction due to differences in electrical charges, attraction of their protoplast. This is the correct answer, this is the correct one, attraction of their protoplast. Check the last one. Cell becomes variable in morphology and function in different regions of the embryo. The process called, that we know that the set of cells they become one organ, the set of cells they become one organ. So they conduct some kinds of functions. So they are differentiated. This particular phenomenon is known as differentiation. Differentiation is the right answer here. Right, this is the third question.